Thank you again for being here today. Hello, everybody. My name is Rosie Albarran-Zecker, and I will be your moderator this afternoon. Welcome to Scripps Research Fully Interactive Resources for Students and Teachers, or FIRST. If this is the first time that you hear about Scripps Research or visit us, I wanted to tell you a little bit about us. So we are one of the largest nonprofit private biomedical research institutes in the nation. And our mission is to develop the next generation of medicines and diagnostics to address the most present health concerns in the world, such as COVID-19. And again, if you're visiting us for the first time, I want to encourage you that after the webinar, you visit our website, or if you have any questions, feel free to reach out to us. Our information is going to be there at the end. We are very proud that we can continue to support our community of educators and students who normally we support in person, but now we're doing this via these webinars, and they're being very successful. So again, thank you for supporting us. Today, you will be meeting Paige Dickson. She's a grad student in the laboratory of Dr. Kudatek, and she's going to be talking about DNA. Her title, her presentation title is Extracting Genetic Material, Playing with Strawberry DNA. And she also will talk about her journey into science and her research work. She will be answering some questions live. Before we go into her presentation and the question and answer session, I wanted to touch base on some housekeeping items. As a reminder, all of you have been muted and your videos have been disabled. The only way that you can communicate with Paige and myself is through the Q&A feature that you're gonna see at the bottom of your screen. So if you hover at the bottom of your screen, you're gonna see a menu and there's a Q&A button. You want to use these to type in all your questions. You can type them in at any time during the presentation, but we're only going to be taking your questions on the second half of the presentation. We also want to interact with you, and we're gonna be presenting some polls. And I think this is a good opportunity now to practice that. So the first set of questions are going to show on your screen. They're right now on your screen. And this one is to get to know our audience. And this is going to inform Paige on how to answer the questions appropriately. So please take a second to answer these questions, which I will share the results live. So I see that most of you are answering the question. Thank you. I think that for most of our audience is high school students, and most of you like science. That's awesome. And what you would like to hear more is about the stories about scientists getting advice on pursuing science. You clearly want to watch this experiment. That's great. And asking about questions on chemistry. So that's awesome. Thank you so much for sharing. And now, without further delay, I'm gonna turn it over to Paige.
Awesome. Thank you so much, Rosie, for that great introduction, and hello to everyone attending. My name is Paige Dickson, and I'm a fifth-year graduate student in Tom Kodatek's lab at Scripps Research. My research focuses on using chemistry to improve the disease states of a variety of different ailments that affect humans, including cancers such as multiple myeloma. I'm happy to talk more about my research after our demonstration, but first I want to get into the exciting part of today. So before we actually start extracting the DNA from strawberries, I want to take some time and just go over the basic concepts of cells and DNA for everyone so that we're all familiar with what we're going to be doing during our extraction. So first, I'm sure you've all heard of the three letters DNA, but what does it stand for? DNA stands for deoxyribonucleic acid, which is the genetic material that contains instructions for how to make a living organism. Each cell in our body and in every single living organism contains a set of instructions for how to exist and survive. This set of instructions can be called that organism's genome. Each cell in our body contains a special compartment called the nucleus, which contains all of the DNA instructions for making that specific cell. DNA is packaged into structures called chromosomes, which are larger bundles of genetic material. Now, let's take a quick pause for a poll. I want to ask all of you if you know how many copies of our chromosomes each of the human cells in our body has. Rosie's going to put that poll up and we'll see what everyone's answers are.
Yeah, so a poll is going to show up on your screen. There you go. If you guys can answer this question really quickly. And it seems that the majority of votes are in for 23. That's what is.
Interesting. That was a kind of a trick answer because so we actually have 23 chromosomes, but we have 2 copies of those chromosomes, making 46 in each of our cells. We get 1 copy from our mom and 1 copy from our dad to make 2 copies per cell. But everyone that answered 23 is correct in the sense that there are 23 distinct chromosomes in each cell that has 2 copies. And what's interesting today about our extraction with strawberries is that strawberries actually have 8 copies of their chromosomes within each cell, which means that they have a lot more DNA per cell than humans. And that will allow us to generate a lot of material to actually visualize this DNA as we extract it. And so as we're trying to get the DNA out of our strawberry cells, we need to break open these cells and gain access to the nucleus. One more fact I want to cover before we get into the demonstration is the difference between plant and animal cells. That's very important for the activity today. Now, I'm sure most of you have learned that there are key features that are both similar and different between plant and animal cells. For example, since they are both living organisms, they both contain a nuclei, or plural for nucleus, that contain their DNA or genetic material. Now, some of the features that differ between these is, for example, the presence of chloroplasts in plant cells, which are the factories of photosynthesis producing their energy. Since animal cells have a different way of producing energy, they do not have these chloroplasts. But for the purpose of the activity today, the key difference between these two is the fact that plant cells have both a cell wall and a cell membrane, protecting the inner contents of the cell, whereas an animal cell only has a cell membrane. You can imagine if we're trying to get into the nucleus of these cells where the DNA exists, then we're going to want to break down both the cell wall and the cell membrane. We will use 2 distinct features of our protocol today to do this, to serve this function. So now I want to go over the 3 key steps for isolating DNA from our strawberries. So the first step is going to be actually breaking these cells open. And the way we're going to do this is in a process that scientists call lysis, which really just means to break open a membrane. And we're going to be using salty, soapy water detergent solution for this step. After we break our cells open and release the DNA from inside the cells, we're going to separate the cell membrane and wall and the solid parts of the cell from the liquid which contains our DNA. And then finally, we're going to precipitate or solidify the DNA using rubbing alcohol, which is a common antiseptic that I'm sure you've had put on cuts before and it's burned. But what this will do is actually cause the DNA to solidify because it is not happy in that type of environment. So we're going to do these 3 steps, but first we need to go over the materials that we're going to use to conduct this experiment. So the first ingredient is, of course, the source of our genetic material, or the strawberries. So I have here 2 frozen but thawed strawberries. You can also use fresh strawberries for this. I've put this in a Ziploc bag, which will give us a contained environment to extract all of the DNA. And then we will need the solution, which will allow those cells to burst open or lyse. And that is our cell lysis solution. Before we go any further, I want to get into a little detail about what the cell lysis solution is, because this is a very important step of our process. So just to break down what this 3-word phrase means, a cell, of course, is the kind of smallest entity of life. And as I mentioned previously, lysis is the breaking down of a cell membrane. And a solution is a dissolved mixture of 2 or more substances. So our cell lysis solution is comprised of a 1/4 cup of dish soap or detergent, we used Dawn, 4 cups of water, and 2 teaspoons of salt, or as we in the lab call it, sodium chloride. I mixed this together until it dissolved. You can see the bubbles from the soap, and that detergent is going to be very important for bursting our cells open. And the reason why this is so important for gaining access to the inside of our cell is based on what the cell membrane is actually composed of. So the cell membrane is made up of a biological oil or fat known as a phospholipid. These molecules have a hydrophobic or water-fearing tail and a hydrophilic or water-loving head. These lipids can interact with each other so that their hydrophobic or water-fearing tails are protected from the water-based environment of our bodies. This tail-to-tail-like composition forms the ring with which our cell membrane is made. Cell membranes are also known as lipid bilayers, bi meaning two, and lipid being the type of molecule that this is made of. Now, if we think about a process that we normally use soap for, washing dishes and breaking through fat and oil, it's not surprising that because our cell membrane is made up of fat-like molecules, that these detergents can interact with the fat-like lipid bilayer and break it down, which ultimately gains us access to the DNA inside of our cell. So now let's continue with our ingredient list, or our material list. So I also have one funnel lined with a moist coffee filter. The moist coffee filter will allow liquid to drain through faster and speed up our extraction. This vessel will allow us to separate the solids in the cell wall from the liquid of our extraction, which contains our DNA. And then finally, our precipitation solution, or the rubbing alcohol, and one toothpick, which will allow us to actually fish out that solid DNA after we've conducted our last step. So now with our strawberries in the Ziploc bag, I'm gonna take my 20 milliliters of cell lysis solution. I'm gonna add it to this bag. We're gonna seal off the bag. And we're gonna use some mechanical force, AKA our hand squishing these strawberries, to help break up the strawberry and also to break down the cell wall. So the detergent in our lysis solution is going to help to dissolve the cell membrane. But as I mentioned before, plant cells also have a pesky cell wall we need to break down. So I'm using mechanical force or squeezing to help break down that cell wall and gain us access to the DNA inside of each of these strawberry cells. So the longer you press and the more you break down this strawberry mixture, the more DNA you're going to get out. So if you spend a good amount of time on this and really break up all of those chunks, you can see I'm starting to turn this into kind of a more homogeneous or evenly dispersed mixture. And once I feel pretty happy about how much I've broken those strawberries down, I'm going to take my filtration device, I'm going to put it inside a clear collection vessel. The collection vessel being clear is important because that will allow us to then see in real time our DNA turning into a solid. So I'm going to add the extraction mixture straight into this coffee filter lined funnel, and we're going to let the liquid slowly drip through. And this is allowing us, again, to separate the solids, which will remain at the top in the coffee filter, from the liquid, which contains our DNA in solution. This is the longest step of the process, and it definitely takes some time. So I've gone ahead and pre-filtered some of the strawberry extract while that one is going. And so what we're going to do is take our ice cold rubbing alcohol. And the rubbing alcohol being ice cold is important because that will help to speed up the precipitation process. I'm going to take this rubbing alcohol. I'm going to very slowly pour it down the side of my container. If I pour too fast, I will disrupt the liquid at the bottom, which will prevent the DNA precipitation from being visible. So I'm very slowly adding the ice cold alcohol to the top. So we're seeing kind of two layers form. So our isopropanol is in the top layer. And we can start to see the formation of this white cloudy material at the interface between our strawberry extract and the isopropanol at the top. And this is our DNA turning into a solid. I can take my toothpick and I can swirl it around in there, and I can actually pull that DNA out. So that is a giant glob of strawberry DNA that we were just able to extract, just with the addition of isopropanol to a mixture of strawberries and soapy water. So this process is very similar to what we do in the lab when we're trying to extract DNA from relevant organisms like human cancer cells. However, of course, we're using slightly different materials than what we use today. However, the process and the concept is still exactly the same. And so with that, I hope I convinced you that you too can extract strawberry DNA, or really any living organism's DNA, with simple things at home that you probably already have. And this is what your finished product should look like. And I've already described to you what the alcohol does. So I hope you enjoy, and I hope you get the chance to do this at home. And I'm happy to take any questions about the demonstration as well as my experience as a graduate student. Thank you all.
Thank you, Paige. That was awesome. So I want to remind everybody that at this point, you can type in your questions using the Q&A button at the bottom of your screen. And I think we're going to have our second poll also coming up, just to gauge how well we're doing. So for this second set of questions is, what are you enjoying the most? And what would you like to hear more about in the second part of the presentation? So I see that everybody is putting in their answers. I think most people enjoyed the experiment. Thank you, Paige, and also listening to science concepts. And there's a mix of things that they would like to hear about. Science careers, if possible, why studying DNA is important.
Okay.
And yeah, and advice pursuing science. There's a mixture of things, so there's a range of things that we can talk about. Thank you. And yeah, go ahead.
Sorry to interrupt, Rosie. Maybe while some questions roll into the Q&A, I can start by just telling you all briefly about kind of my story and how I ended up here at Scripps Research. So growing up, I was always really fascinated by the concept of human biology. My dad is a doctor and my mom also is a medical professional. So I was always surrounded by this concept of kind of human health and disease. And at first I thought I wanted to be a medical doctor, but I quickly realized after working in retail for a year in high school that I did not want to be around people as much as I did, or at least sick people. However, I still loved the idea of science, and I wanted to contribute to kind of solving human disease in a different way. And when I took chemistry and biology in high school, I really fell in love with the different laboratory activities that we got to do in those classes. I still remember working with flies, which is a classic model organism, in my senior year of high school. And from there, I moved on to major in chemistry at Northeastern University with the support of one of my high school teachers, who was my first mentor in science. And at Northeastern, I had the opportunity to do an internship at a pharmaceutical company called Bristol Myers Squibb. And there I was really surrounded by this pharmaceutical industry, and I loved the experience. And I knew I wanted to continue studying science, and I didn't want to go to work immediately. So I looked into PhD programs where I could continue studying human health and disease at the molecular level. And I joined Tom's lab 5 years ago. And during my time there, I have been involved in the identification of new chemical matter that could be potentially used as drugs in the future for diseases like multiple myeloma. I've also been involved in a project that was identifying diagnostic antibodies, or the part of our immune system that is responsible for recognizing foreign matter. And I use that in the context of looking for antibodies for dengue and Zika virus, which are two well-known and very ubiquitous viruses across Earth. And you can imagine that these types of diagnostic approaches will also be very useful for the current pandemic, COVID, and I'm sure we've all heard about these antibody tests And so that's kind of one aspect of the science that I've been working on over the past 5 years, which has been very exciting to now see become very relevant. And after this, I'm not exactly sure what I'm going to end up doing. I love research and I still love working in the lab, but I'm also very passionate about education and communication. And there are a variety of different careers that involve science that can serve those purposes. So I'm still figuring that out. It's never too late to figure out what you want to do in life. So you have plenty of time for all of you that are on.
Great. So we have our first question here, and it's how different are the solutions used for animal cells for lysis and precipitation?
Awesome. Great question. So for precipitation, it is absolutely identical because human DNA, plant DNA, all DNA is made up of the 4 same bases, A, C, T, G. And so once you get the DNA in that extracted state, you'll use the same type of alcohol no matter what type of cell. Now, for animal cells actually gaining access to the DNA, it's actually a little simpler than what we did here. So because they don't have the cell wall, you don't require as much mechanical force to break open the cells, but you still use the same detergent and that same principle of breaking down the lipid bilayer, no matter if it's animal or plant cells. Great question.
Great. So I want to remind everybody to type in their questions, and we actually made it in a way that if they want to do it anonymously, you can. I know some people are shy, so we have turned this option on, and maybe that will increase the number of questions. Meanwhile, what would you say that is your advice if any of the students watching us today are really passionate about science, but they don't know if their future career lies in research or not? So how to start?
So my biggest suggestion is twofold. One, talking to people who are in jobs that you think you may like, whether that's professors who do research, people at local pharmaceutical companies. I would encourage you all to attend networking events, which allows you, well, right now it's a little harder because of the coronavirus, but there are a lot of virtual platforms to engage with people in professions you may be interested in. So for example, LinkedIn. It's never too soon to get your LinkedIn up and running. But more formal programs such as internships will allow you the opportunity to have built-in mentors in a given industry that you may be interested in. So my experience in undergrad at Bristol Myers Squibb was very motivating, and it really set me on the trajectory that ended me at Scripps here today. For high schoolers, there are a variety of summer internship programs. Scripps Research offers one, and I think other local universities and institutes do as well. And this is a great opportunity to get full-time experience doing research and also being exposed to people who are later on in their career in that field. Outside of research, there are also plenty of internship opportunities in other science-based tracks and careers, such as policy and journalism. So I encourage you all to look at some of the national organizations like the AAAS, AAAS, and they have a lot of different internship opportunities as well. So I think, talking to people, exploring your resources on the web, and doing science is really the best way to know if it's something that's for you.
Great. So we have a question about your research. So I'm not sure if the exact word is the one that they use here, but how do you extract cancer cells, or how do you get cancer cells for your studies?
So there are several different ways. So there are two types of I'd say, flavors of cancer cell, if you will. So one is a primary cell, which is sourced from a human tissue sample. So this is basically if medical doctors go in and biopsy a tumor from a patient who has cancer, you can take that tumor out and actually break it down to the single cell level and work with it in the laboratory. Now those cells, because you've taken them out of their biological environment, do not grow infinitely. They don't have the capacity to replicate outside of the body. So sometimes we can take these primary cells and turn them into continuous cell culture models. And this typically involves changing their genetics so that they are able to replicate infinitely. So typically, by and large, all of the cells we're working with are sourced from a human, whether they've been modified afterwards or not, depends on what our purposes are. Typically, we're working with immortalized cell lines, which means that they've been treated in a way that can replicate forever. And that just makes it a lot easier and more reproducible for us to do our science.
Great. So a question on what classes you should take if you are interested in science, and also a little bit on how do you know what to study? How do you know what to pursue?
That's a great question. Yeah, I'm still figuring that out. There are so many. So, when I was in high school, the classes I took were biology, chemistry, and physics, right? That's kind of the way we think about science branches. But then you get into these subspecialties, which are so very different. So in biology, you can have molecular biology, which is kind of what I'm doing, looking at the single cell level under a microscope. But then you can also have systems biology or ecology. And these are looking at more of an organismal or even larger level at the way life exists. So I would say, really, again, reading and learning about all of the different types of science just through, different classes. Start with a basic chemistry, a basic biology, a general, take all of your general courses. And if there are certain topics or kind of studies or types of experiments that interest you, continue to push further with that. If you are in college, talk to your teachers about the different opportunities that are available. There are an endless number of fields of research and topics that you can study at the intersection of what would seem to be totally opposing fields. So, there is science going on in every realm. So it's hard to kind of suggest how best to figure that out. It's very personal, and I think it just takes more experience. But I would say take as many different classes as you can, because there are things that you may not actually think will interest you that could be very cool, neuroscience, or like I said, environmental science, and a variety of other things.
Great. So there's another question on how scientists share their information, or how you can get information from other scientists, and if projects are kept private.
So typically the way that research works in academia or at universities, or at places like Scripps, is that we do research and we do not share that information until we've kind of derived conclusions from the experiments that we've done. And the way that we do this is in the form of scientific publications or research articles. So once we've written up our experiments in this research article format, which is basically like a very long lab report with a lot more information, then that is available to all other researchers. Now, not all research that gets done gets published. So there is a variety of studies that may not actually make it out into the public sphere. But what I will say is there's an increasing push right now for open access to the research that goes on in labs. And there are a bunch of different movements, such as the different archive websites that are now available for putting information into the public sphere. Pharmaceutical companies and industry tend to share information less, simply because for them, sharing information could be the difference between one of their competitors taking that and potentially usurping their profits on a drug. So there are slightly different approaches. Now, that's not to say there's not collaboration in industry as well, but it's just very different purposes for the research that goes on in academia and industry.
Great. So our next question is, do you work with genetic engineering? So I'm not sure if the person also could have meant genetic engineering. So maybe you can approach it both ways, if we have engineers in biomedical research, and also how much of what your work is genetic engineering.
Okay, so I will say that engineering is just, I view engineering as creating solutions, for problems that you encounter, right? So being a scientist, you are inherently engineering solutions for problems that come up in lab, whether it is technically not having the right piece of equipment that you need to do what you want to, or whether it's designing a new system to test a new question. So engineering, definitely, that concept is kind of a basis of a lot of the research and science that goes on in the lab. And genetic engineering, my interpretation of genetic engineering is anything that manipulates a cell or an organism on the DNA level. And so for me personally, I do genetic engineering. For example, when I'm trying to study how a specific protein in a cell is involved in that cell's growth or survival, I will genetically engineer that cell to not produce that protein. And then that allows us to study whether or not that protein is important for the processes that I'm looking at. So there are a lot of different genetic engineering tools. I'm sure some of you have heard of CRISPR, which is becoming very popular now. And these are definitely things that we're using in the lab, and I personally have as well.
Great, that was a great question and answer too. So can you tell us a little bit about your typical day at work?
Well, every day is different. So, experiments oftentimes, they run the course of several days, and each day you're doing something very different. So for example, if I was extracting DNA in the lab today, I may want to put that DNA into a host cell through a process called transformation. So I will actually put that DNA, genetic material, into another organism. Typically, we're using bacteria. So that process will usually take a day, and then I may want to grow those bacteria up. So today, if I'm extracting DNA, tomorrow I may be growing bacterial cells. The day after, I may be extracting the protein that is produced from the DNA that I put into those bacterial cells. But oftentimes, the day plays out such as in the morning, I'm planning experiments and making sure that I have everything I need for the next few days. And then I actually get into the lab and I do those experiments. The amount of time that I'm spending in the lab varies on the experiment. And then usually towards the end of the day and in any breaks, I'm analyzing data, reading papers, potentially going to meetings, and doing kind of other non-bench work. So it's definitely a testament to time management and being able to balance multiple obligations and also being very organized. So I would say the days vary, but that's also the exciting part, is that you're never doing the same thing twice, maybe twice, but actually 3 times for statistical significance. But every day is different, and that's very exciting about being a scientist.
I guess that I can ask, do all the experiments work out the way that you plan them?
Well, no, they don't. And that is also the fun. Sometimes experiments fail for reasons that are totally out of our control, like the coronavirus. The machine breaks, something happens, the water bath wasn't at the right temperature. You have to get used to kind of rolling with the punches in that way. And also the fact that we're doing research to answer questions that people don't know the answers to means that sometimes those answers are not what we expect. And that actually sometimes comes to be the most interesting science that we can do. So a lot of the time experiments will either fail in the sense that you don't get a result, or you don't get the result you expect out of them. But that just allows you to start asking more questions and really continue the research on.
Great. And I know that you already talked about this, but there's still one question. What advice do you give to students that are thinking about getting into research, and how do you actually begin working in a laboratory? So maybe you can focus your answer just on that step.
Yes, absolutely. So how to find a laboratory. I would start at your university. If you're a college student in your university, every department website will have a list of professors that do research, and you can either email them, or if you know them personally, make a physical introduction and open a dialogue about your interest in research. Perhaps the first question you ask is not, hey, can I work in your lab? But ask the question you just asked me. I'm interested in research and I would like to get involved. Do you know of any opportunities that are available to people like me? And most of the time, professors or people in academia are very willing to help, and they want to guide you to where you want to be in your career as well. And so I would say talking to those people, whether you know them from classes or you find them on a website. If you're not at a university, do the same thing for a local university that's in your area. If you're in Jupiter, Florida, look up Florida Atlantic University or Palm Beach State College. They don't have research, but Florida Atlantic University or Scripps. If you're in San Diego, look for UCSD or one of these other universities, and just start that way. And I would also advise you, if you start in a research lab and you decide that things are not exactly what you expected, don't be discouraged from all research. I did 4 different internships, or I worked in 4 different labs during my undergraduate studies. All 4 of them were so very different, but they all fell into the realm of biomedical research. So every lab has different techniques and experiments and questions that they're asking, and every person is different with what excites them. So the more people that you talk to, especially on the professor level, they'll be able to give you some insight into where or if they actually have research opportunities for you.
Great. And there's a comment here if, like you mentioned, one of the colleges, state colleges, doesn't have research going on, you can always, even if you attend that college, you can always reach out to other colleges that do have research and opportunities. And also, you can look for ways of transferring to colleges that have research opportunity if that's something that you realize you are passionate about.
That's a very good point, Rosie, because I did my bachelor's degree at Northeastern University in Boston. And ultimately, the lab I did most of my undergraduate research in was actually at MIT. And the way that I got into that lab was simply emailing the professor, telling him I loved the science he was doing, I was really passionate about it, and I wanted an experience in his lab. And fortunately, there was space and he was willing to take me on. But really, you don't know what opportunities are available until you ask. And the sooner you start exploring those, the more you'll realize there's a world of different possibilities for you in terms of training experiences.
Great. So I know that we're almost reaching, the 40-minute, which, we wanted to keep this at the 45-minute time frame, and we don't have any other questions. So I guess, this is your last opportunity to type in any other questions if you like. And if not, I'll ask Paige, what's the take-home message? Do you have one take-home message for all the students?
My take-home message is find something you're passionate about and pursue it with great force. That was really, it has propelled me from step to step in my career. I can't tell you exactly what I'm going to end up doing as a job, but I can tell you that I love science, I'm passionate about research, and I'm passionate about teaching. And those things will have really propelled me throughout my career. So find the things you enjoy and the work will come. And you will feel the most rewarded from that. Especially because science itself is very hard. And if you're not passionate and you don't enjoy what you do, it can get very exhausting. So make sure you're doing things that you enjoy, but that are also meaningful for the world.
Great, that's awesome advice. So now we have the third and final poll. This is gonna help us figure out how we did and how we can improve in the future. If you can take a second to answer these questions. Meanwhile, I'd like to tell you a little bit about our upcoming webinars. So next Tuesday, we have a panel of graduate students who are going to be discussing issues in science advocacy. So I invite you to join us. It's at the same time. And next Thursday, we don't have an experiment, but a science explanation by another of our graduate students on ribosomes and their role in health and disease. And she's also going to talk a little bit about her project. So if you're interested in learning more about, stories of scientific projects that are going on at Scripps Research, please join us for those next two webinars. And thank you for everybody who put in their answers. I'm excited that you liked the science demonstration today and that you're motivated and that you understood. That's great. And if you don't mind, when we close this Zoom call, also you're gonna be, there's gonna show a survey on your screen, and we appreciate if you can take that survey as well. There's gonna be a place in that survey where you can talk about potential topics that you would like to hear on future webinars. We plan to continue having these webinars for the end of this semester and starting again in the fall. So please give us as many topics as you want to hear about. I know that our grad students and postdocs and even our faculty are excited about sharing their stories with you. And with that, I'd like to thank you all for attending today, and I'll see you next time.